Today, I'm going to talk about some thoughts and considerations that I use when doing binary classification for disease outcomes using human biobank data. So I'm going to particularly focus on dementia. And I have done some work using the UK biobank, which is one of many biobanks. And UK biobank has about 500,000 people in it. And what kinds of data sources might you want to use to try and predict who will be diagnosed with dementia? So there are three specific data sets that I have used. One is a proteomics data set, which comes from blood plasma. Another is a neuroimaging data set, which comes from various brain imaging modalities like MRI and diffusion tensor imaging and others. And finally, there is a cognitive tests data set, which looks at measures of affect, as well as things like numerical and spatial reasoning. So when you look at complex data sources like these to try and predict disease, you also want to use good baselines. And when I say baseline, I don't mean using logistic regression and comparing that to a deep learning classifier. I mean baseline sets of features. So very basic clinical and demographic characteristics that you would want to check against your more complex, harder to obtain data sources. So when we talk about baselines, that can include things like demographics. And that can include things like age, sex, education, maybe ethnicity, and ApoE genotype, which is the most well-characterized genetic predisposition to Alzheimer's disease. Now, in addition to these very basic demographic factors, you might also want to look at clinical risk factors. So that includes things like smoking or alcohol consumption or history of head injury. And it's good to be very comprehensive with baselines because you want to be very rigorous when justifying the use of more complex and costly data sources. So these are the different features that you might want to use to predict dementia. Now let's talk about how we actually use them. So let's talk about data splitting. So when you split a data set to run a machine learning experiment, some people, they might do something like a random 80-20 split for training and test, or maybe a stratified 80-20 split, or you might do something like 60-20-20 for training, validation, and test. Here is something that I did. So this is my very bad drawing of the UK. And in the UK Biobank, there are all these different assessment centers where the data in the UK Biobank are collected. So what I did is I grouped them into 10 geographical regions. And I used a geographical cross-validation procedure where one of these regions is held out, and then you train on the other 9, and then you iterate over all 10 of these regions, performing the same procedure. So that's how data splitting was done. And now let's talk about the model briefly. So I use an algorithm called light gradient boosted machine, or light GBM. And what's really nice about light GBM is that it scales really well to high dimensional data sets with lots of samples and lots of features. And it's very fast in comparison to other kinds of models like random forests or XGBoost or even histogram gradient boosting classifiers. And whenever you have a model like this, you have to optimize the hyperparameters. And so for that, I used an auto machine learning framework called FLAML, which stands for Fast and Lightweight Auto Machine Learning. And the thing that FLAML takes advantage of is that during your training procedure, it's very easy to reduce your error when you're early in the training procedure. It becomes more costly and time-consuming to reduce your error when you're later in the training procedure. And so FLAML takes advantage of this by exploiting lower cost sets of hyperparameters earlier in the training procedure before moving on to more complex and costly sets of hyperparameters in the search space later in the training procedure. Okay, so we have this cross-validation procedure and we have this modeling approach. Now let's talk about metrics. How do we evaluate these models? Now, what too many papers do is they draw a ROC curve with the true positive rate on the y-axis, false positive rate on the x-axis. You have your chance along the diagonal. You draw a curve, and maybe your AUC is 0.8 to 0.9, and then you declare victory and you go home. And one of the take-homes of this talk is that this is bad. Do not do this for multiple reasons. One reason is that when you calculate the AUC of a ROC curve, you are considering decision thresholds that are absolutely nonsensical. So for example, the last point of a ROC curve corresponds to a decision threshold of 0. In this case, meaning that it predicts that everybody has dementia. So your true positive rate is going to be 1, which is great, but your false positive rate is also going to be 1. Not great. So do you really want to consider decision thresholds that are very far on the x-axis corresponding to high false positive rates? I wouldn't. I don't think you would either. So one thing that you can do is to do a partial AUC corresponding to the part of the curve that encompasses decision thresholds that are clinically or biologically reasonable. That's one thing you can do that's good. Another thing that you can do that's good is to actually pick a decision threshold. So when people use packages like scikit-learn, the default decision threshold is 0.5. Why should we use 0.5? The answer is you shouldn't, and you should have some kind of rational procedure for choosing a decision threshold. And there are many different procedures that people use. One is called the Luttens J statistic, which is the binary case of a more general metric called the bookmaker informedness. And it's pretty simple. It's the sensitivity plus the specificity minus 1. And so you ride your ROC curve and you see which threshold corresponds to the highest Luttens J statistic, and now you've chosen a specific decision threshold. And once you have a specific decision threshold, well, now you can draw out your confusion matrix with your true positives, true negatives, false positives, and false negatives. You have your actual cases and controls, your predicted cases and controls, positive, negative, positive, negative. Now, there are many, many metrics that are threshold-based that you can then calculate from your confusion matrix. And one that I want to draw your attention to is precision. And precision is also known as the positive predictive value. And this is corresponding to the true positives divided by the predicted positives. And the reason why precision is so important is that in medical contexts, we are almost always dealing with highly imbalanced data. It's usually a small minority of your data set that is part of the positive class, such, e.g. having the condition that you're trying to predict. And when you have a high class imbalance, this ROC curve can be very, very misleading. So I'll give you a little worked out example. Let's say your sensitivity, which is also your true positive rate, is 0.9, and your specificity is also 0.9. And these are characteristics of your model or your test or your predictor. But precision is also dependent on prevalence. So let's say only 5% of the people are in the positive class. So you can rewrite precision as a function of these three quantities, and you can do it like this. So PPV is your sensitivity times your prevalence divided by the same thing, sensitivity times prevalence, plus 1 minus your specificity times 1 minus your prevalence. So if you work this out, what you end up with is 0.045 divided by 0.045 plus 0.095, which is about 0.32. So again, Precision answers the question, out of who you predict is positive, who's positive? And the answer is 32% in this particular context because of the low prevalence, which means 68% of the time, when the model predicts that somebody has dementia, they don't, which is why prevalence is so important. Now, the last thing I want to talk about is one particular summary metric that is very rarely reported, but is a really convenient summary of a confusion matrix. So you can use a confusion matrix to calculate rates that we're all probably pretty familiar with. So when you divide by the actual number of positives, you get the true positive rate. Divide by the actual number of negatives, the true negative rate, so on and so forth. Now, when you divide by the predicted number of positives and negatives, here again, you get the positive predictive value. When you divide the true negatives by the predicted negatives, you get the negative predictive value. When you divide the false positives by the predicted positives, you get the false discovery rate. And when you divide the false negatives by the predicted negatives, you get the false omission rate. And now there's one metric that actually synthesizes all 8 of these metrics into one number, and it's called the Matthews correlation coefficient. And it's essentially the positive predictive value times the true positive rate times the true negative rate times the negative predictive value, square root, and then you subtract the false discovery rate times the false positive rate times the false negative rate, finally times the false omission rate. And so this is just one very convenient metric that can enable researchers to more transparently report their results in clinical machine learning, in those cases where you often have a highly imbalanced data set, in which case the ROC curve can be very misleading. So in some, use rigorous baseline sets of features to try and justify the use of complex data sources, which might be very costly to obtain. LightGBM is a scalable, very fast algorithm. Auto machine learning packages like Flamel can take the work out of optimizing hyperparameters. And transparent, comprehensive metrics reporting is extremely important and should be done regularly. Often I see precision values that are either never reported or they're relegated to the supplements of papers, and it's a very serious problem. And that's it.
So Bayesian optimization is a paradigm for optimizing black box functions. So maximizing or minimizing a black box function f. Now, the thing with f is it's hidden, it's latent, and we don't know the ground truth. We don't know its analytical form. And neither, so we don't also have access to its gradients. The only thing we have access to is a few very expensive evaluations of f. So another thing about f is not only is it hidden or latent, but it's very expensive to evaluate. So the big caveat in BayesOpt is that you have a budget that you can evaluate f only m times. And m is potentially a very small number. So you want to optimize this black box function f, and the only thing you have access to is maybe a few evaluations of f. So you have where you evaluated f and the evaluation, and this is expensive, right? So one can think of this as sort of some setting where, you know, f of x is like the free energy perturbation of a molecule or some kind of in vivo trial outcome. So something that's really expensive, and you want to get an idea of where f is maximized or minimized only by using this tool of like you can only evaluate f m times. And so Bayesian optimization is sort of a methodology, a general purpose methodology that you can kind of use to sort of optimize functions f, which are sort of a black box. So the analogy that's actually used is, so imagine, you know, this is 1800s and you're on like a patch of finite land in Australia, and you know that there's gold in 2 spots, and you can only like dig m times, and you need to basically find gold or increase your probabilistic sort of probability of finding gold. So how are you going to go about doing it? Obviously, randomly digging in spots is not going to be a good idea. So I'm going to just like basically cast this into like a 1D problem, because I think that's the easiest to understand. So imagine that this is my latent ground truth. Right? And I obviously don't know this, but for the purposes of illustration. And my points, the 2 points whose expensive evaluations have access to, kind of live here and here. So the way the algorithm works is that as a first step, I would fit a surrogate model, a probabilistic surrogate model to my 2 points. And the paradigm that I'm going to use is a Gaussian process. You can use other surrogate models to kind of fit these 2 points, but I think it's going to become clear why you actually need a probabilistic model to fit these 2 points. So this is basically just simple nonlinear regression setting. You have 2 points, you have x and f of x, and just fit a smooth function through it, right? So I'm going to use a Gaussian process, and I'm going to fit a smooth sort of curve through these 2 points. So I'm going to get something like this, potentially, right? And I'm assuming that they're noiseless, but of course, these points might have noise. So the setting is epsilon is IID noise. And the thing with the Gaussian process, so F is modeled as a Gaussian process. And I'm not gonna talk a lot about how to fit a Gaussian process, but I'm gonna say a little bit about it. I mean, you don't actually need to understand how a Gaussian process fit to data to kind of understand Bayesian optimization, but you have great software to fit a Gaussian process. And the thing about Gaussian process, say, versus neural networks is that a Gaussian process is a probabilistic nonlinear way of fitting a data, right, in a regression setting. So what you get when you fit a Gaussian process is not just a single function estimate, but what you get access to is actually a whole space of functions compatible with the data. So you actually get a probability distribution of curves or functions that are compatible with the data. So I'm going to just draw this band here. Which really symbolizes that there are actually loads of functions in this space that are going to be compatible with the data. And these are, you can think of these as like maybe 95% confidence intervals, right? So in a Gaussian process, so just a detour, you have a prior, which is a prior over a space of smooth functions. And now the smoothness of the functions is controlled by the choice of the kernel function. And then when you fit it to data, so if you observe 2 data points here, the space of functions collapses around the data. So then you get functions that look like this. So they pass through the data, but they vary a lot where you don't see any data. So you see data here, and you don't see data there. So the uncertainty bands around your data obviously collapse, and then they kind of go, the uncertainty bands kind of widen out where you don't have data. So this is a very, very actually compelling property of using Gaussian processes, is that they give you this uncertainty quantification by design. You get that for free when you fit a Gaussian process to data, right? So a Gaussian process is the surrogate that we're going to use to model our kind of expensive valuations. But then why do we need this probabilistic paradigm? Like, what are we going to do with the uncertainty there? So now comes like the key part. We're going to actually use the uncertainty to decide where to sample next, right? So the next thing that we do in Bayes-Opp, so the first step is we fit a surrogate probabilistic model to your expensive data points. Like here, I just have 2, but you can have like, maybe more in a real setting. The next step is you compute something called an acquisition function, right? Which is also something that you compute on your entire sort of input space X. And the acquisition function is something that is derived from the probability distribution that you fit to your data. So in the most simplest setting, one can just look at the uncertainty, right? So what happens to the uncertainty in the input region after you fit your surrogate probabilistic model? You can just plot it, right? So this would look, you can just compute it and plot it. It just kind of goes up here, right? So I have no data, so the uncertainty potentially just is very high in this region. And the important thing is fitting a Gaussian process is actually quite expensive. So you can only do it to very few, you can only fit a GP to like very few data points. When I say few, I mean you can go up like few thousands. That's because the computational complexity of fitting a function f using a Gaussian process is actually n cube. And the memory, and it's quadratic in the memory complexity. So computing the acquisition function is cheap. Fitting a Gaussian process is non-trivial, that's expensive, but fitting an acquisition, deriving, writing out the acquisition, computing the acquisition value across your input spaces is very cheap. So that you should be able to do cheaply, and you can do it across the whole sort of input domain. So you kind of derive this acquisition function, so which is like a scoring function. It just indicates to you where you're going to sample next, right? So you just look at the acquisition function, this 1D case, and be like, okay, I mean, it seems like this is a good point because that's where my score is maximized. And here the acquisition function is just a proxy for the variance, right? So assuming like this is basically, the width of your uncertainty, it's a proxy for the width of your uncertainty interval. And then you can just say, okay, this is where I'm going to place my next sample. So this is where I'm going to evaluate X3, right? And then the algorithm sort of proceeds. So this is basically one loop of your Bayes-opt procedure. And now your data set has 3 evaluations. Okay. And as one can imagine, so I need to just replicate the same function. Okay. Point here, have a point here, and then I evaluate somewhere here. And then again, I sort of fit my surrogate Gaussian process to my 3 expensive evaluations, and I get something like this. Starting to get a lot better, right? So this is basically a primer, like a really sort of kind of vanilla Bayes-opt application in the one-dimensional case. So you kind of do the same thing. You fit your surrogate GP to your very expensive evaluations. You compute your acquisition function, and then you find out where you're going to sample next, and then you add that to your data point, and then that's how the algorithm proceeds. So it kind of alternates between these 2 steps. There's a lot of rich theory behind acquisition functions, right? And I'm not going to have time to kind of go into the details of, several of them, but one very important concept in choosing good acquisition functions is this concept called exploration-exploitation trade-off. So if you didn't have the acquisition function and you just had access to the surrogate model, you can say, okay, I think one strategy is exploit, which means just sample aggressively over the points where you know the ground truth, hoping that maybe you're gonna uncover something. Or the other strategy, just exploration, is that just sample where the uncertainty is maximized. So that's what we're doing in this acquisition function here, which is just a proxy for the variance around the predictions, right? And the variance is basically how much the space of functions actually vary in that region where you don't have the data. But a really good acquisition function will have that trade-off. So neither will it just look at, neither would it tell you to just aggressively sample around a point that you already know the ground truth of, and neither would it just tell you to just go for explore, which is just basically just sample somewhere where which is farthest away from any of the points that you've uncovered. So an example of an acquisition function that's used looks something like this. So it's got this lambda parameter that controls this trade-off. And so all of the acquisition functions in literature that are actually popular have this kind of trade-off built into them. So I'm going to kind of switch tones a little bit for the last few minutes to talk about how Bayesian optimization can be used in a generative model. Oh, 2 minutes, right? So this is going to be really quick. So of course, this was just a contrived example. This is 1D You can also do Bayesian optimization in the batch case, where instead of just sampling one point, you're doing like a batch of points. But in the case of generative models, assume you have a generative model for small molecules, right? So you have like a VAE-like setup, and you have a latent space, which itself, I mean, it's supposed to be low dimensional, but let's say it has about 10 or 15 dimensions, and you have a decoder, and say, you know, what you're actually encoding. So this is the VAE sort of autoencoder setup. This is the latent space. And so each molecule that you train with this VAE setup is represented as a compressed point in this low dimensional space, this low dimensional latent space, right? So each point here represents a point in your training data, and assume that these are like, you know, small molecules, so a drug modality, and you only have access to some really expensive property, like some kind of maybe a sort of free energy perturbation space using Bayes opt, such that maybe you want to sort of maximize or minimize this property, right? So you only have access to these 2 points. What you can do is essentially fit a Gaussian process to this high dimensional setup. So this is a 1D example, but you can fit a Gaussian process to any dimensional data and essentially do the same thing. So instead of like fitting a Gaussian, this would be essentially fitting a Gaussian process to a high dimensional sort of surface, and then using kind of the same machinery here to navigate the latent space of this generative model to keep then coming up with proposals that you can then decode to get your small molecule of interest, right? So this is how essentially Bayes opt is actually used in modern day science. A lot of the times it's not just used to optimize some black box function like in a vanilla setting, but a lot of times it's actually used, it's embedded as an auxiliary algorithm in the latent space of generative models to actually come up with interesting proposals where you're kind of maximizing or minimizing a certain property of interest.
The problem that we're interested in is how do cells compartmentalize proteins? And by compartmentalizing proteins into different compartments, and there is a variety of these. Some of these are membrane bound, like the nucleus. Some of them are contained within that compartment. And I'm going to highlight 2 here. This is the nucleolus. I'm drawing it quite large here. And then you can also have compartments called transcriptional condensates. And both these function in different physiological processes. So they need to concentrate different types of molecules to do their function. And what we know is that these compartments have different proteins that can be concentrated in them. There's different types of scaffolding molecules that accumulate in there. And they're not all different. So some of them, there can be an overlap between these proteins. But we want to understand what is the chemistry that makes them distinct. And protein language models have provided us the ability to now go ahead and do this. And the work that I'm going to describe to you is an example of how one might. So we took 12 compartments throughout the cell. This includes diverse physiological processes such as gene expression, the compartmentalization of stress, cognition. All of these processes transcend through these different bodies. We have about 5,000 or so protein sequences. And we can use this information as input. So actually what we did specifically is we took ESM2, and with ESM2, we have a representation of protein sequences that we can generate for each and every one of these. So take ESM2, this model, we feed it in our protein sequences, and we get out a big matrix, n by 320 in this case. So n is the length of that protein sequence, and we can then sum this up and get an embedding. And this will give us a vector that we can then go and use to fine-tune ESM2 to understand what is different between all of these different compartments. So we add a couple of additional layers onto our model, onto this large language model. And now, when we feed it an unknown protein sequence, it will spit out the probability of a protein being concentrated in one of the 12 compartments that we looked at. And what we found is that this actually works pretty dang well. So for our test set, the prediction probabilities were pretty accurate. Great. So that to us is something of, at least we're getting at some of the chemical information inside of protein sequences that lead them to be compartmentalized in different parts of the cell. And what we wanted to do was then go and demonstrate this. So we want to take this ability to figure out whether or not a particular peptide is associated with one compartment or another compartment. We wanted to use this as a tool to actually make something that we can go put into a cell and look at using a fancy microscope and see if it's going into the right compartment or the wrong compartment or what's happening. So what we did was we took this model, and by the way, I should have said this earlier, but we decided to call it ProGPS. It's sort of a GPS for your protein. And the idea was to take this ProGPS and think about ways that we could take a protein. So proteins can have different sorts of properties to it. One type of domain on a protein is a folded domain, and another is a disordered domain or an intrinsically disordered region. And this is a structure that doesn't really adopt a certain one really highly occupied structure. So it'll sample a lot of different states. So it's very dynamic, it's changing. They're actually incredibly useful in biology for doing an array of different functions. And what we wanted to do was add on a region onto a protein sequence that then would cause it to go into a specific compartment. So our first approach was to use a greedy search-based algorithm to try and model this process. So we actually failed spectacularly with this one. But it worked in the sense that when we looked at the data, the experimental data, we could actually see the proteins were moving around. So we tapped into some of this information. And but there was problems. And some of the problems were things like 13 tryptophan residues in a row. That's bad. You will cause aggregation, all kinds of things like this. And so we knew we had some of the right sauce, but we weren't quite there yet. So in our second approach, and these are all sort of generative strategies for testing protein codes, really. So in our second strategy, we did a Markov chain Monte Carlo blocked Gibbs sampling procedure. And so I'm going to tell you a little bit about the energy function that we used. And I think that this approach really encapsulates a variety of different approaches for sampling chemical space and protein sequences that is critical for getting something to the right place. So as a whole, this looks something like, and this is building really upon the work that Reeves and coworkers put out. So there's a projection term. And so this refers to the ability of a protein amino acid sequence to match a particular backbone. We have a language model term. So what that really is this protein sequence that we're sampling found in nature? And ESM2, if you're not familiar, is trained on 60 million protein sequences. So it learns something about that. The other terms in here convert as sort of a prior. So this is an n-gram version where we have different small sets of amino acids and asking how does our protein sequence we're sampling match that. And the innovation here that we added in was two different things. So we have ProGPS, and this component of model tells us to, well, what we're going to do is sample subsequences of proteins or sequences of proteins that are found in the target compartment. So we'll sample chemistry that you might actually find in that specific condensate. And the other term that we have here is one that enforces disorder. And the disorder term, so going back to this, right, we're adding on a new domain, and the sequences we want to sample are going to be, disordered fractions of this, or are these components, and we want to enforce a disorder condition, so that this fraction of the protein has a higher propensity to be disordered, and therefore less likely to influence the structure of the folded domain or influence the other properties of the protein. It's more of a sort of a tuning of that protein. So when we did this, what we found was that depending on how litigious you'd like to be about this, we could get between 10 and 100% performance out the small subset of sequences that we looked at. So, we went back to the nucleolus, and this is a great example. It's a very large condensate, and so it assembles material inside of it, and it's very easy to observe. So experimentally, most of the sequences that we got into this one, 10 out of 10 were a technical win, in that when we compared it to a control protein, all of them were doing better than that control. And then we started looking at these other smaller bodies, and what we found was actually quite interesting. We were able to get partitioning into these, but they're not all the same. So there's something stochastic about the way these things assemble into different condensates, and the material that accumulates in them is not always going to be the same between one compartment or another. So when we went and trained this model on all of this data, we were getting different flavors of different condensates. So we understand that the imperfections in this model and our approach is now kind of a relationship about, well, how good is that data? How good is that the approach that we took to experimentally build a model of each of these compartments? Okay, so we could test these things out experimentally. I think we learned quite a bit about how to generate proteins using a fine-tuned model like this, and things to consider. The chemical space of proteins was really bottlenecked in this strategy. We were thinking about protein sequences that are found in nature. We were thinking about the types of sequences that are specific to that compartment. And then we were also thinking about protein sequences and features that are associated with a specific type of domain that wouldn't alter the overall structure of that protein. And if I have time, the last thing I'll tell you about is how we then took this tool, ProGPS, and started asking questions about, well, if you have a protein sequence and you have its mutated form that's found in disease, how are those two things different? And how is the information content in the wild type protein different from the information content in the mutant protein? And can we use that to learn something about how nature manifests information that drives the distribution of proteins? And so this is what we did. We took something like, I don't know, we've actually ran something like 300,000 different proteins that are found in disease and have an uncertain prediction or a benign characteristic, but they're all mutated. And we asked, we took the wild type and the mutant, and we pushed this through this algorithm. What is the distance of the distribution of probabilities for the wild type and mutant? So we do the Wasserstein distance of this, and what we found is that if you look at the data for this, there's something of a histogram that you could draw, where there are some that have no effect, essentially. So you're mutating some part of the protein that doesn't change its distribution. Okay, I completely buy that. That's actually an expectation, right? If you mutate a catalytic residue, then perhaps you shouldn't alter the distribution of protein. Maybe you should. But if you mutate something on the surface or something that's important for the fold of a protein, and now that protein is opened up like a flower in the springtime, then that's going to be able to interact with many different parts of the cell. And so that surface area is exposed and changes the distribution of the protein. So absolutely, a lot of these are going to alter the distribution. And what we found is that for pathogenic mutants, we could then go and experimentally investigate these guys, and we would see really dramatic differences between what we would find for the wild type protein and the mutant protein. So they really are moving around. And what are the things that are important for this? It's actually quite incredible that you have the same magnitude of effect coming from the folded domain, from the IDR domain. So you could have something that is just a sort of a disordered conformational ensemble of states that has a similar degree of effect that something on the folded domain can have. This is really possible because we were able to, or all of these findings are really possible because we were able to take the data from many different protein sequences and learn all of the connections between the folded and disordered domains simultaneously. We weren't pigeonholed into what biochemists have been doing for the last 100 years or so, which is looking at individual motifs and pieces of proteins. We can learn all of the relationships at once using these transformer models. And I think it's a powerful tool for now moving forward to understand, how can we build small molecules and proteins that are going to go to the right place in the cell and help them optimize their therapeutic activity.
Today, I'd like to tell you a little bit about AlphaFlow, which is a deep learning method that we've developed to try to model protein conformational ensembles given a protein sequence. So to set the context, you've likely all heard of AlphaFold, which addresses the protein structure prediction problem. Given an input sequence, it produces the most representative output structure for that sequence. So pictorially, we can think of proteins as living in these in this dual space, a sequence space and a structure space. And throughout, please forgive my terrible protein drawings. And AlphaFold has learned a mapping from this space to that. Now, of course, this is a slight simplification of reality, as Henry already alluded to in his last talk. Proteins do not adopt a single structure, rather an ensemble of structures, depending on the binding partners, the molecular environment, the pH, the ionic strength, and of course, just regular thermal fluctuations that cause atoms to constantly move and jiggle around as in intrinsically disordered regions or proteins. So instead of a single well-defined value for this function, we actually have a distribution of structures, possibly with multiple modes. And AlphaFold has done an excellent job of learning a representative structure among this distribution. So you might think of AlphaFold as learning something like a mapping from the sequence to the expectation of the structure conditioned on this sequence. Now what we would like to do is to learn a mapping from the sequence to the entire distribution of structure given sequence in a way that we can then sample for new sequences at inference time. So this is what we want. And AlphaFold has not been trained or formulated for this task. So what we do in this work is we extend AlphaFold under a generative modeling framework called flow matching to model these ensembles. So flow matching is the algorithmic framework that makes this possible. And in flow matching, we have two distributions, P0, which is going to be some noisy prior distribution, for example, an easy to sample Gaussian. In our case, it might be some arbitrarily defined distribution of unfolded structures. And then P1 is going to be the data distribution that we want to sample. And for now, we're just going to think of the space that these distributions are over to be some Euclidean space R3n, where n is the number of atoms in the protein. Now, this is a slight simplification of what's actually presented in the paper, but it gets the main point across. And what flow matching provides is a means to learn a time-dependent vector field, which we're going to call V. It's a function of xt, which is going to be like intermediate values that interpolate between the prior and the data. It's going to learn a flow over those values that pushes the distribution P0 to P1. And this gives us a generative model, because by construction, we can easily sample points from the prior, and then we learn the flow that allows us to evolve those points to P1. So pictorially, we can draw this as having the prior on the left-hand side, for example, and let's say it's just Gaussian. And then on the right-hand side, we have P1, which is some more complicated data distribution. And again, this will be our distribution of like unfolded protein structures, and this is our distribution of folded protein structures. And we want to learn a flow that pushes this distribution to that one. And in doing so, it creates an evolving set of intermediate distributions, which we'll call PT of xt. And flow matching enables us to learn this flow in 3 steps. So in the first step, we define a noising distribution, or we define a way of adding noise to some given data point x1 to get intermediate distributions xt. Right? So this is a family of distributions indexed by time and indexed by x1, such that at time equals 1, this distribution is just saying, don't add any noise to x1, so it's over here. And at time t equals 0, it adds so much noise that it's equivalent to the prior. So pictorially, we can maybe take as an example just some like linear interpolation between a given data point x1 and the prior p0. And in this interpolation, then the way that you would sample xt given x1 is by sampling the noisy point x0 and then just linearly interpolating between the two. So for example, we sample some point x0, then we interpolate to x1, and then we choose whatever time t we're interested in, and we get xt. So this is how we add noise to the data. That's what we defined. Second, we define a denoising vector field that actually undoes this process. So this denoising vector field is a vector field that for a given point x1, so right now we assume we know what x1 we're interested in, it pushes p0 to x1. And we're going to call this denoising vector field u of xt given x1. Now, in this linear interpolation example, this is also very easy to define. This is just the vector field that says, whatever point I'm currently at, the direction that I should go is in the direction of x1. And I'm going to do so at a rate that allows me to get to x1 in the remaining time, which is 1 minus t. Now, this is what we've defined so far. And you can sort of see maybe where this is going. We have these vector fields that push the prior p0 to any individual point x1. And what flow matching tells us is that by marginalizing over x1 in the appropriate fashion, we can learn a vector field that pushes p0 to the marginal of x1, which is p1. And so that's the third step. We're going to learn, typically, and in this case with a neural network, some v of xt given t and theta. This is now a neural network. And this is going to be the expectation of this denoising vector field, where this expectation is conditioned on the clean data point, given that we are currently at the noisy data point. So let me just unpack this a little bit more operationally. So what we do is we sample points x1 from our data, and then we add noise to those data points by doing this interpolation. And in doing so, we get to some point xt. But when we're at a particular xt, there were actually many possible clean data points that could have gotten us there. Right? So here we could have done this interpolation. We could have also sampled x0 here and x1 here and done this interpolation and arrived at the same point xt. So in these two cases, we have two different values for this denoising vector field u, one pointing in this way and one pointing this way. And now what flow matching tells us to do is to learn the expectation over all of these possible interpolations that went through xt of this denoising vector field, which will generally point somewhere in the middle. And just to hopefully make things a little bit clearer later, we can actually write this through a little bit. And we find that this denoising vector field can be parameterized by a denoising model, which is just predicting what the most likely data point is, given that we're currently at the noisy data point. And then we do some appropriate transformation to incrementally step towards that. So in our protein setting, like I said, this is going to be our distribution of unfolded protein structures. This is the distribution of folded protein structures for some sequence of interest. And what we've now defined is a way of adding noise to these protein structures. And we want to learn, given a noisy protein structure, what was the most likely noiseless protein structure that gave rise to that noisy protein structure. So that's where AlphaFold comes into the picture. We're going to use AlphaFold to parameterize this denoising model here. And I apologize, probably should use different colored markers for this sort of thing. But flow matching tells us that we need a neural network that predicts the expected denoised protein structure and has this noisy protein structure as input. So how do we do that with a model like AlphaFold? So AlphaFold right now is taking in the sequence as input and the MSA. And it's roughly predicting, as we said earlier, the expectation of the structure given that sequence. And now we just need to modify AlphaFold to also take as input the noisy structure so that the prediction is now the expectation of the structure given the sequence and the noisy structure. Now, how do we do this? So recall that AlphaFold has what's called a template track as input. So in addition to the sequence and the MSA, AlphaFold also accepts as input a template protein structure, which can help it inform its output protein structure. Now, typically the template input it's not always used because it doesn't turn out to affect the accuracy of the final prediction that much. But in this work, we actually use this fact to our advantage and leverage the template input, not to provide a template, but to provide a noisy structure, the noisy structure xt. And this provides us with a way of training AlphaFold under this flow matching framework so that it actually learns to push a distribution of unfolded structures to an ensemble of folded structures rather than just a single one. And a conceptual way of understanding why this works is that in the absence of any noisy input, the sequence input is completely deterministic. So the output must also be deterministic. AlphaFold can only ever produce a single structure. Again, a bit of a simplification here, but it produces a single structure, and so it has to pick one that's representative of this entire distribution. But now by having the noisy structure, we inject some stochasticity into the system. So AlphaFold can look at what the current noisy structure is and observe that it contains hints about which of the many possible ground truth structures this noisy structure came from. And so AlphaFold can use that to affect what the output structure is. So it's no longer just a deterministic function of the input. In summary, what we've done is we've provided a way of making a relatively minor change to the AlphaFold architecture, right? Because template stack's already there, the entire architecture is unchanged, the training laws is unchanged because we're still learning an expectation, so it's still the same regression-based loss, frame-aligned point error loss. But now we have a principal method for learning from protein ensemble data, right? So right now that might mean data from MD simulations. Of course, relative to static protein structures, we still have a relative dearth of dynamic ensembles in the public data sets. But hopefully, as that increases in the coming years, methods like this can help us better take advantage of those data and use existing foundation models like AlphaFold to model these more richer output labels that more accurately describe the protein in vivo.
So what we want to do here now is no longer generate protein structures, but now we want to generate DNA sequences. So I'll just write down one, a few letters. And we want to generate sequences like this. So now we have these discrete objects, right? No longer our continuous protein structures. And we have like a sequence of these discrete categories. And the way that we'll represent the sequence is just via like a sequence of simplices. Or what's simplices? If we now consider, or let's assume we have only 3 letters in our DNA sequences, of course, we will have 4 letters later on, but in all my drawings, we will consider having 3 letters, then our sequence of simplices we could draw it down like this, and then say, oh yeah, our letter A here is represented as this corner of the simplex, our letter G as this corner of the simplex, and C as this corner of the simplex. And then what we want to do is to generate like this the sequence, and then at the end we can decode this sequence of simplices into a sequence of nucleotides and a DNA sequence. Yeah, and then for this flow matching process, right? Bowen told us we need this prior here. Where was it? His P0, and we have our data distribution, the P1. And now I'll always only talk about a single simplex because then it's easier and I need to draw this. And if we now look at our single simplex, then what could our like it's clear what our data is, right? For example, if for our single simplex the data point is this, it's an A for the simplex, then we would have this corner as our data point on it, and our what we could choose as prior. Oh yeah, as prior is, for example, a uniform distribution over all the simplex. And yeah, now we want to define our flow matching procedure for that to generate this data like this A or these corners. And of course, later on, we'll stitch all of this together, right? All of our simplices and our neural network, it will always be one that takes all these simplices as input, then predicts an output for all the simplexes jointly. And it won't be as primitive as just orienting where you want to flow mass on a single simplex. But now to see how we do this on a simplex, or how we define our flow matching procedure on the simplex, let's consider again these 3 main quantities that Bowen wrote down here. So this conditional probability path, then this conditional flow map or this interpolant, and then this conditional vector field here. And these three objects are connected with each other, right? You can go from the conditional flow map to the vector field by differentiating it. And for example, this vector field is connected to the conditional probability path by the continuity equation. Just saying that if we have this, or what is the vector field that gives rise to this conditional probability path? Yeah, and now let's just do what Bowen did here again, just with a different P1 and a different P0, and do this what we call the interpolant perspective, where we first define this interpolant here, and then trivially, just via differentiation, like we differentiated it and got this guy here, our conditional vector field arises. We train our neural network to just match this conditional vector field, and then we end up because we train with many of these x1s, with many of these data points, we end up with a vector field that approximates the non-conditional vector field, that approximates this guy. And yeah, if we now do this for a simplex, let's think about what happens. And I mean, it won't be the final solution. And final solution. And now we have our data point here, right? Let's say we sampled A as our x1, and then we sample somewhere on here from our uniform prior, right? Our x0. And then our x1 with this linear interpolant here, with the t going from 0 to 1, it'll be somewhere on this line. So maybe if our t is 0.5, then it'll be here in the middle, this xt. Then Bowen told us we take the derivative of this interpolant, we get this little vector, conditional vector field here, we match our neural network against it. Okay, then this works, and this is something that we can do. But let us look at what is the conditional probability path that arises if we do this procedure. Okay, for that we will probably draw a few more triangles. And so let's look at the conditional probability path at time 0.5. So conditioned on this x1 here, which are the possible points from this probability distribution, conditioned on this x1, what are the possible xt's that we could have reached? Well, and it turns out that the possible xt's are in this triangle here. And it's a uniform distribution over this triangle. Why is that? Because consider what is the furthest away point that you could sample from your prior, your uniform prior. It is somewhere down here at this face of our simplex. And then at time 0.5, this point would go here. And you could also have uniformly sampled another point from the prior, right? This one right here, then the point would end up here, and so on and so forth. But all your points, they will end up in here. Okay, then why do I think, I didn't say it yet, but this is a problem that we have this conditional probability path that does not at all times t, like here, for example, the one with time 0.5, that does not at all times t have full support over the space that we're interested in, and not full support over the entire simplex. And why does this happen here and not for Bowen's continuous stuff? Well, because here we have a compact space, a bounded space. And in Bowen's scenario, right, you could have always had a point that's like much arbitrarily far away because you have this Gaussian. And here you have a boundary and you can't get arbitrarily far away, so to say. Okay, now why do I think this might be a problem? Well, let's draw another triangle. We have this a lot here today. And then let's consider the conditional vector field of this point and this point at time the 0.8, right? At time 0.8, this guy's conditional probability path will look something like this. And this guy's conditional probability path will look something like this. Okay, and then let's draw the conditional vector fields that Bowen was drawing here, right? We have all these, this guy. So we have all these interpolants going from data to our point that we condition on. Sorry, going from x0 to x1, so from noise to data. And then let's say this is our xt here. Then our vector field will be pointing to this corner. And the vector field will always be pointing to this corner. And of course, this conditional vector field over here will also be pointing to the corner. But now, if your data could have come from here or from here, then the vector field marginalized over these two here, or the change of the vector field in which direction it is pointing here, it changes discontinuously once you cross this border here, right? And also in terms of, so this vector field is discontinuous in space, the marginal vector field. And presumably, if we want to approximate something with neural networks, then having something like discontinuous is probably not so nice. And we also in our experiments show that where we have like a smoothly changing vector field in space, this seems to work better. But now, what do we actually do? And what do we do instead of this, what we call the interpolant perspective, where we first define an interpolant, this thing here, then take the derivative and very easily get our training target. What we say is, right, with this, we arise with these, or we obtain these conditional probability paths that have this non-desirable property that it does not have full support over the entire simplex. So instead, we say, let's start by our flow matching procedure or our flow matching design by defining a conditional probability path. So this is now what we want to design first, and then all the other stuff follows from that. And then, okay, what choice could we make? The choice that we make is, if this is again the point that we condition on, then we want a sequence of conditional probability paths that in the beginning, right, it should be a uniform over the entire simplex, and at the end we want it to be a Dirac at our data point. Okay, and then what we choose for this is just a Dirichlet distribution, where this alpha parameter here, it increases, or as time in our goes from 0 to 1. So as t goes from 0 to 1, or this alpha parameter will go from 1 to essentially infinity, where infinity is 8 because that's far enough. And then these three parameters will correspond to, if up here is our data point, is that here will be the ones for these two corners, and up here will be this alpha parameter. And then the way this looks like, well, maybe we don't all know how these Dirichlet distributions look like. It is just that if this is maybe 1, if this is 1, then this will be the uniform distribution. If this is 2, then like a lot more mass is contracted up here than a lot more mass is up here than down here. And it just goes on and on like this. And if you have like 8, for example, as alpha, then almost all mass is up here and almost no mass is down here. Okay, so now we have just written down, oh, we would like a conditional probability path like this, instead of conditional probability paths like the red one. And then we still don't have a training procedure. So to obtain this training procedure, we then now need to, instead of just taking the derivative, as we did here, we need to, we have a conditional vector field defined, take the derivative, we have our interpolant defined, take the derivative and get the conditional vector field. And then we can match against this conditional vector field. Here we have the conditional probability path, and we said it is connected to the conditional vector field via the continuity equation. So now we defined this, this conditional probability path, PT. Let's write it down, PT. And then the continuity equation just tells us that it's the change in density with respect to time. This should be the same as the divergence of the vector field. So it just tells us that if we have a point here, then the amount or the change of density in time should be the same as the amount of density that basically flows into this point and out of this point, or the sum of that at that time point and at that point in space. Yeah, and then we go ahead and solve this continuity equation, which there's an interesting trick to do this, where we just look at the flux across a plane that goes through this point, because all the all the density along this plane will be is the same in this Dirichlet path, and we obtain this vector field, and that is what we match against. Yeah, and then to summarize, we tried doing this interpolant perspective for defining a flow matching process on the simplex, had some or found some issues with doing that, and then instead decided to take this probability path perspective, and this then gives rise to some complications for finding your training target. But in the case of this simplex and the Dirichlet probability path, this was or this is possible to find this vector field. And I think maybe this is the interesting takeaway. If we have like a space that, for example, it's a manifold with like a cut locus or some compact space, like our simplex here, for example, then it might be interesting to take a different perspective on flow matching, this path, probability path perspective, instead of this interpolant.
Thanks to the organization for the invitation for me to present a very light talk on how we have used deep learning to navigate vast chemical spaces to find novel therapeutics for a wide variety of pathologies. So with this, I would want to introduce how traditional methods have been doing ligand discovery campaigns. And an example that's still widely used, certainly here at the Broad Institute, would be high throughput screening campaigns. So this would be a system where you have a very large chemical library, which is stored in plates. And at the Broad, it's around 800,000 individual unique molecules, but has now been expanded recently to contain over 1 million molecules. So what you would do is that you would, for instance, in the context of an antibacterial discovery campaign, you would have in your experiment a vial, or in the well, you would have your bacteria here floating around. And then you're basically going to test individually each molecule in your high throughput screening library for its antibacterial properties. So you're checking with the phenotypic readout whether the bacteria dies or doesn't die upon exposure of your small molecule. So what you're then trying to find is basically, if you would have done it in multiple concentrations, you're checking for molecules that are active or inactive. And using this procedure, and now more recently in a more automated or using robots version, you can actually experimentally validate a large chunk of molecules, but it's still limited to what you actually have physically accessible in your library, which is around, the best case, several 100,000 molecules. So the chemical space is quite narrow if you consider that the total drug-like chemical space has been estimated to be 10 to the power 60 molecules. So another way of doing things is going computational rather than doing an experimental high throughput screen. So what you can do, and this is with recent advancements in combinatorial chemistry, mainly driven by the Ukrainian chemical vendor Enamine, is if you would have on the shelf a bunch of building blocks. Building block A, building block B, building block C, and you would do an in silico enumeration of these building blocks to afford virtual molecules. You could end up with, in this case, most recent library sizes are now up to, I think it's 50 billion molecules that are so-called make-on-demand molecules. What's special about these molecules is that they can be readily synthesized in 4 weeks, and the vendor has a synthesis success rate of up to 80%. So these molecules don't exist, but they are in catalogs that you can access. So 50 billion molecules, acquiring all of these and testing them individually is completely intractable. So now we need to rely on computational models to help us prioritize specific molecules for synthesis and experimental evaluation. So what can we do now? We can actually use the data that you have gotten through a high throughput screen. So again, you have your library with all your molecules, and then basically based on the experimental phenotypic readout, you can classify the molecules, or you can binarize them into active molecules or inactive molecules. So you have a very large data set, like in our case, to 40,000 molecules of actives and inactive molecules. And now you can train a neural network here, which in our case was a message passing neural network. And then you can now look in this very large chemical space of billions of molecules using your trained neural network to find the compounds that have the highest predicted antibacterial score. And then you could have molecules that have a like score of being antibacterial, which can be low or high, and then you prioritize the molecules that are top scoring. You contact the chemical synthesis vendor, and you prioritize those molecules for synthesis, and then you explicitly evaluate them in the lab. And if you have lots of data and lots of molecules, you can actually deconvolute the molecules that you have. For instance, in this case, you could, using a Monte Carlo tree search, basically find a region of the molecule that's most contributing to the antibacterial score. And this could then be considered something like the explainable AI realm. So then in this case, the annoying thing is that even if you do find molecules that are antibacterial in the experiment, you still don't really know what their actual mechanism of action is, or what they're doing inside the cell, or what protein they're modulating. So if you find a molecule, which would then be denoted as a hit molecule, and you want to optimize the physical chemical properties to make it more like a preclinical drug candidate, if you do not know what protein target your molecule is modulating, you're basically optimizing in the dark, and you need to rely on brute force medicinal chemistry efforts to find a better molecule. So, and with this, I want to introduce another biological system that we have been working on in the lab recently, which would be so-called dinoflagellates, which are marine plankton. So these molecules, they biosynthesize a specific molecule called brevetoxin. And brevetoxin, for the sake of time, I'm not going to explicitly draw out here. It's a very large molecule. Basically, what it does is that in our neuronal cells, we have specific, this would be the phospholipid bilayer. We have specific proteins that are involved with the management of ion flux. One of these would be the voltage-gated sodium channel. And brevetoxin, if I denote this as B, lies on the inside of this ion channel and keeps it open. So you would basically have a net influx of a lot of these sodium ions inside, which would mess up the cell signaling in the neuronal cells and can lead to all kinds of pathologies. So what we then did is the same thing as here. We got a library of 2,500 molecules, and we have individually in each of these wells, we had the neuronal cell and a small molecule and the brevetoxin, and our readout is basically which of these molecules rescue the neuronal cells from dying. And then based on that, we can again train a neural network. And we have used the Broad 800K library in this case and found 100 molecules that had good scores. And then we've tested them experimentally in the lab to see which one of these actually rescued the cells. And a nice molecule that we found here was, let's see if I draw this correctly. This molecule looks like this actually. And what's so interesting about this molecule is that it actually contains this motif here, which is nicotine. So again, in the lines of explainable AI, if you think about this substructure and about nicotine itself, is that the nicotine is also involved in the sodium ion management, also in neuronal cells, and then nicotine acetylcholine receptors. They look like this. They're pentameric structures. And basically, you have alpha and beta subunits, and depending on the stoichiometry, you have a different isoform of the nicotine acetylcholine receptor. And nicotine itself lies in between an alpha and beta subunit, and then basically either through agonism or antagonism would also modulate ion flux. So this could be a potential mechanism of action to rescue neuronal cells. So then what we've done is that we looked again in the Broad 800K library for molecules that contain a nicotine substructure and that had a high score from our neural network. And 11 of the tested molecules all had nanomolar potencies in rescuing the neuronal cells from the marine toxin. So that doesn't mean that if you are exposed to shellfish poisoning that you need to start smoking, but it is a nice indication that this chemotype might be leading to a specific mechanism of action. So now that we have a specific protein structure in mind, this pentameric structure, there are experimental X-ray and cryo-EM structures of the nicotine acetylcholine receptor. What we can do now is use these very large chemical spaces and rely on instead of from a ligand-based discovery campaign, we can move to a structure-based drug discovery campaign and basically build molecular docking models of these subunits, and then use the scoring function of this molecular docking model to navigate this very large chemical space of billions of molecules, and in conjunction with our neural network, look for compounds that have a good interaction energy and a good phenotypic learned score for inhibiting this marine toxin. So what we have done in the first step is that we looked for molecules in this billion space that contain the nicotine substructure. And now we've docked them against this system. But what we're also trying to do is find molecules that do not have a nicotine substructure and see if we can find an entire new scaffold that would also exhibit the cell rescue phenotype. So with this, I hope I was able to convince you that there's many ways of using machine learning to navigate very large chemical spaces, and that there's multiple layers where you can use machine learning. So we can also use AlphaFold or the AlphaFlow, as was mentioned earlier, to create ensembles of these structures. We have also built neural networks that were trained on cytotoxicity of normal cells. That could be an additional filter for these billions of molecules to enrich for molecules that would not be cytotoxic, but would still rescue the neuronal cells from the marine toxin. So yeah, this is a very much ongoing project, and we're very curious to see where we end up.